Hi Bio30. This lesson is 16.3, part 2, looking at what could go wrong when meiosis has division of chromosomes that does not end up dividing them equally. So normal meiosis in human cells starts out in the testes or ovaries, and a cell with 46 chromosomes in the human ends up making gametes, in this case, in this picture, after meiosis 1 and meiosis 2 are finished, we end up with 4 sperm cells that would have 23 chromosomes in each one. Our lesson today is going to be about nondisjunction, which is, instead of disjoining when anaphase happens, anaphase 1 or anaphase 2, when either the two homologous pairs are supposed to, pair of chromosomes are supposed to separate, or when in meiosis 2, the actual, over here, when the chromosome separates at the centromere, when one of those two events does not happen as planned, we can have a wrong chromosome number in our resulting gamete, which then means if that was the gamete that got fertilized, you could end up with genetic disorders in the offspring. So that is our topic today. We're going to start where we left off on recombination. First of all, just a little couple slides talking about how the recombination of genes can happen. And first of all, we look at if there wasn't any crossing over, just the fact that there's always a randomness of when cell division happens, the mom's gamete and dad's gamete will have, from the mother, she could have number 1 from her dad, 2 from her mom, number 3 from her mom. You just never know which chromosomes are found in that gamete. So the gametes have a mixture of, for any particular gamete, let's say if it was a human sperm cell, it could have, from the guy who made it, it could have number 1, 2, 7, 10 from his mother, and then 3, 4, 5, 6, 11 from his father. And so you never know what combination of chromosomes end up in that gamete, and so it is very random. So they say that if you have with 23 pairs of chromosomes, you'd be looking at mathematically 2 times the 23rd of how many different combinations you could get with 23 pairs of chromosomes, and 2 to the 23rd is actually like over 8 million different combinations that a man could have of sperm, of combinations of some chromosomes coming from his mom, some from his dad. And so he would have a guy would have 8,388,608 different sperm formed. However, that's without thinking about the fact that the chromosomes, remember, crossed over. So when prophase 1 was happening, these chromosomes were crossing over and swapping genes. So there are this many possible sperm cells. However, because crossing over can happen, there's basically an infinite number of different combinations of genes that could be found in sperm. That's pretty crazy to think about. So they mention here that in prophase 1, this is where synapsis, or the homologous pair, comes together, and this is where the non-sister chromatids will cross over. What that means is it's not the chromatids that are of the same chromosome that cross over. It is actually the ones that belong to, this one is maybe one from the mom, and this is the one from the dad. And so it's this is like number 6 from mom, number 6 from dad. And so it's the the non-sister chromatids, but both of the same number, that will do the crossing over. And what happens there is, let's say the person making these sperm cells has all dominant genes for these two traits. However, if crossing over was to happen, if this was this one here was the chromosome that ended up being fertilized, you notice it now has a dominant and a recessive for those two traits. So maybe this was like for a plant that's tall, the other gene was short. Maybe this is for round seeds, the other one was wrinkled seeds, or whatever it is. The combination is new. So in what we started off with is one parent had all dominant genes here, big A, big B. One parent had all recessive genes, small a, small b. But because of crossing over, they have some resulting gametes that have a combination of some dominant, some recessive. So you can see how there's a mix now found any one chromosome in a sperm cell or an egg cell. Any one chromosome that you look at, like let's say chromosome 10, there's already some genes in there are from mom and some on there are from dad because of crossing over. So this is a microscope picture of a homologous pair crossing over, and you can see that the crossing over is happening in many locations, not just one. And so it gives you a lot of different genes from mom and from dad in one chromosome. So what we're going to go to next is what happens if the meiosis process doesn't happen perfectly. And they say here that either when the homologous pair separates in anaphase 1, or when the chromosome splits at the centromere and the two sister chromatids separate at anaphase 2, in either of those cases, if separation doesn't go perfectly, we could end up with an extra chromosome in the sperm cell or egg cell. And so what they're showing you here is that in meiosis 1, this homologous pair is supposed to go its separate ways. And you can see what happened here is that they didn't. So maybe there were, for some reason, I don't know, they could have been curled up too much and crossing over or something. But as you can see, 3 of the chromosomes went one way and 1 went the other way. So that would be non-disjunction that happened in meiosis 1. In meiosis 2, the chromosome is supposed to separate from its centromere here. And in meiosis 2, if that happens, in this case, it's just saying that could also cause an extra chromosome in one of the endings resulting gametes and an extra in another resulting gamete. So this is showing you meiosis 1, where the pair went together to one side. And so it's got 2, let's say, number 7s in its gametes, and the other gametes wouldn't have a number 7. So if it happens early on in meiosis 1, it basically messes up all the resulting 4 sperm cells. If it was fine in the first one, in meiosis, the homologous pair separated, so we're okay there. But then in meiosis 2, this one was okay, but this one, the chromosome did not separate. The two parts of it stayed together. And what happens there is that if the problem of non-disjunction, of wrong separation of meiosis happens in meiosis 2, it messes up 2 of the 4 gametes, the other 2 are okay. So the earlier that it happens, the more it messes up the resulting cells, the resulting gametes, and the chromosome number in them. It mentions here that in anaphase 1, what could go wrong is that the homologous pairs don't separate, and what could happen wrong in anaphase 2 is that the sister chromatids that are supposed to separate don't separate. In anaphase 1, it would be the homologous pair that was supposed to separate, doesn't separate. Okay? And what happens is you end up sometimes with a resulting new little zygote that has, instead of 2 of everything, they might have some chromosome that is called monosomy that has 1 of that chromosome number because of some problem that happened in meiosis, and the gamete that was ended up being fertilized had a missing chromosome. Or sometimes the gamete had an extra chromosome because of something like this. And then when we look at the resulting new little baby, it has 3 of a certain chromosome instead of just 2. So, for example, if we look at Down syndrome, you can see that the karyotype here shows that it's got 2 number 1s, 2 number 2s, number 3s, number 4s, number 5s. They get shorter and shorter. When you get to number 21, you see that there are 3 of them. And so what happened there is that either the sperm or the egg had been formed improperly. There was nondisjunction that happened, and either the sperm or the egg, you'll never know, had an extra number 21. And so when the sperm met the egg, there was 1 of everything, but one of the gametes had 2 number 21s. The other one had the 1 number 21 it was supposed to, and you ended up with a trisomy 21, which is 3 of some chromosome. Now, that extra chromosome does cause a lot of problems, as does a missing chromosome. And so this Down syndrome person, as all Down syndrome people would have, is all of their 100 trillion cells in their body would have 47 chromosomes in total instead of 46 chromosomes per cell of their body. So they will all have this extra 21, number 21 chromosome. And so when you have this extra, you end up with some kind of general features that seem to be consistent among Down syndrome individuals. Things like the rounder head and sometimes some mental difficulties and learning problems, some folds to the eyes. There is a range of capabilities and abilities of Down syndrome individuals, but they all do have this trisomy number 21 if you look at their karyotype. And that would be why some genetic tests are done before the babies are born, sometimes with a sample of amniotic fluid or a sample of chorion cells. Now there's even a blood test called the Harmony test that can do a genetic test on cells from the baby to check ahead of time before they're born whether they have a genetic disorder. The second genetic disorder here, Edwards, you can take a look at the karyotype again and see that it's got now trisomy number 18 here, 3 number 18s, and you have 2 of everything else. Here you see an X and a Y, so this is a little male. This one for Down syndrome was a female. You see 2 X's, they're the same length of the last 23rd pair of chromosomes. Here it's a longer one and a shorter one, so you know it's a male. And the trisomy 18 has these symptoms that you see here with misshapen ear and skull and clenched hands and a very short life expectancy. Patau's is a trisomy 13, so either the sperm or the egg had an extra number 13. When the fertilization happened, the resulting cell had an extra 1, and this one, I've got little videos with or links that give you more information about each of these genetic disorders, so you can check those out. But often there's problems with the eyes, cleft palate, again, clenched hands. Turner's, I really suggest you watch this Turner syndrome video. It's excellent, and it shows you a couple of individuals with Turner's. They have basically, with Turner's, they have an X chromosome, but they don't have a second sex chromosome. So they would be considered an X0. And so it is female with female characteristics, but there is problems with their sexual development, and they have a short stature, a broad chest, And those are some of the main features. You can see that in this picture here. Klinefelters would be, I have a karyotype here, would be where there were two X chromosomes, but also a Y. So XXY is a typical Klinefelters, and what happened there is that the chromosome that was extra was the sex chromosome, the 23rd X, and so it ended up giving you this individual that has, so the problem was in the egg cell that the X chromosome stuck together, the two X's stuck together and went together. When the egg was formed, the sperm carried the Y, and so it ended up being XXY, giving you male in anatomy. However, the shape of a female and breast development, and so there's basically some characteristics of males and females. This individual was Erica, but then after winning a women's downhill skiing, ended up not passing the genetic test and was basically told, no, you are actually more male. And so there's an interesting historical story of an Olympic gold medalist who I believe lost their gold medal, because that's kind of what happens with failing genetic tests and Olympics. Okay, we are going to go back to number 6. Triple X, sometimes even more X's, but extra X chromosome instead of having a female with 2 X's. If the genetic disorder is that they have 3 sex chromosomes, it could be metafemale or triple X. And there's, again, menstrual issues. This lady, Iwa Klobukowska, she lost her Olympic medal because they said that the extra X they found that she had when they did a genetic test must have given her the advantage, and that's why she won the medal, so that was taken away from her. A lot of controversy in some of that, but there it is. 2 Ys, XYY is a Jacobs, and that's condition you could see here, does still have the X chromosome and the Y, but has an extra Y. So there's some features that are, you can read about that are characteristic of an extra Y chromosome. The last one is B30 Ch, and this one is when there isn't an extra or missing chromosome, you still have 46 chromosomes, but number 5 has a chunk missing. So it has a deletion on a portion of number 5, and then the baby actually sounds like a crying cat. So you can listen to one of these and check that out for yourself. And there's some other features that are always consistent with a B30 Ch individual. Awesome video, meiosis square dance, check that out. And to wrap up today, just a comment about how making sperm and making eggs is different. I mentioned this in the Google Meet today if you joined us, but when sperm is formed, spermatogenesis is the formation of sperm, and there will be this sequence of events that happens where a spermatogonia, like starting at Go in Monopoly, I think of that when I explain this, but spermatogonia will start off at the edge of the seminiferous tubules. Remember that it's the seminiferous tubules and the testes where spermatogenesis happens, and our spermatogonia would be the cell on the edge, and then meiosis 1 and meiosis 2 will happen here until you get the sperm formed, these little yellow guys with tails, that will be able to swim up the seminiferous tubules and end up in the epididymis to mature. So this is where we're talking about sperm production in the seminiferous tubules of the testes. And the sequence of events there is that the spermatogonia turns into a primary spermatocyte that will undergo meiosis 1, and then it'll turn into a secondary spermatocyte, which will undergo meiosis 2, which will end up with a cell that's a spermatid, which grows a tail. Once it grows a tail, it is finished. It is done. I always think Z is the at the end. It's completed. And the spermatozoa is what we shortened to the word sperm. A spermatozoa is the mature sperm cell. So you can check out this link and check that over again. But again, it is a spermatogonia that turns into a primary spermatocyte that goes into meiosis 1. A secondary spermatocyte then goes into meiosis 2. A spermatid grows a tail and becomes a spermatozoa. So spermatozoa would be these yellow guys here. Okay, so that's the sequence of events. Here's an actual microscope picture of testes, cross-section of testes. Remember, these interstitial cells here in the middle are making testosterone, whereas the seminiferous tubules is where sperm are forming, and it's that spermatogonia that's here, and all these guys are the spermatozoa. This picture, again, just showing you if you had to draw an arrow, spermatogonia is where we're starting, and they're developing until they form spermatozoa over here. Okay, there's another picture of spermatogenesis. And so spermatogenesis, as you know, starts off with one cell with its 46 chromosomes. At spermatogonia would still have 46 chromosomes in human terms, but all four sperm would end up with 23 chromosomes once they're done all of meiosis. Now, the difference is that when eggs are formed, in meiosis 1, first of all, the formation of eggs is happening in the ovary, and it's called oogenesis. But when oogenesis is happening, the difference here is that the first division in meiosis 1, all of the cytoplasm goes to one egg cell, and the other one dies, and it becomes a polar body that has very little cytoplasm and can't survive. So in meiosis 2, we just have the one egg cell left that's going to undergo meiosis 2, and the same thing happens. All the cytoplasm goes to one side, and we get a second polar body. So in the end, we get one large egg cell, that's all, one that is released ovulated from the ovary, whereas sperm, you'll have 4 of them. So whenever you get a question about comparing how sperm are formed compared to how eggs are formed, sperm will have 4 resulting cells from spermatogenesis, and eggs will have 1 resulting cell from oogenesis. And these pictures are showing you the exact same thing. This is showing you the comparison. You guys can check it out, same thing. And this is the same. Oogonium is again starting at go. Primary oocyte will go through meiosis 1, and secondary oocyte will go through meiosis 2, and you'll end up with the egg or ovum that's haploid, 23 chromosomes, but only one of them. Okay. Last little bit on twins, just that if it's fraternal, then that means that there were 2 sperm involved, which means there must have been 2 eggs involved, and they would not be any different than siblings of any sex. So it could be, they would look not identical or anything. If it is identical, it would have been 1 sperm met 1 egg, and in the morula stage, some cell would have broken off, and they would have developed separately into 2 identical babies. And they say that that happens 30% of the time. Cool little link here to check it out. And that wraps up our talk today on non-disjunction and